Okay, so what is Killing Our Fish by yours truly, Amanda Brown? Chemical structures and key functional groups. So first we're going to go over kind of how molecules interact and chemical structures. So what is a chemical structure? The arrangement of chemical bonds between atoms in a molecule, specifically which atoms are chemically bonded to other atoms based on the chemical bond, together with information on geometric shape of the molecule needed to identify the type of molecule. So for example, water consists of H2O, two hydrogen atoms connected with one oxygen atom, and then how molecules interact with each other. So molecule interactions are between molecules or between atoms that are not linked by bonds. Molecular interactions include cohesive attraction between like, adhesive attraction between unlike, and repulsive forces between molecules. Cohesive interaction is an attractive force that exists between molecules of the same substance. For example, rain falls in droplets rather than a fine mist because water has a strong cohesion, which pulls the molecule together, forming droplets. Adhesive interactions are forces between unlike substances. In case of a liquid wetting agent, adhesion causes the liquid to cling to the surface on which it rests. When water is poured onto clean glass, it will spread, forming a thin, uniform layer over the glass surface. This is because the adhesive forces between the water and the glass are strong enough to pull the water molecules out of their spherical formation and hold them against the surface of the glass, avoiding the repulsion between like molecules. Now, repulsive interactions is two charges of like nature that are pulled far from each other. For example, the force between two same-side magnets is repulsive because both the negative charge and positive charge will not connect with the same charge, hence that they repel each other. So eukaryotic cells and overall function of the organism. So this is kind of the main structure of a eukaryotic cell, and they are organisms that contain a nucleus. The eukaryotic cell has a nuclear membrane that surrounds the nucleus, in which the chromosomes, which contain their hereditary material, are located. Eukaryotic cells also contain organelles, like organs, including mitochondria, which contain the cellular energy, a Golgi apparatus, the folded membranes that produce secretions and intercellular transport, an endoplasmic reticulum, it's a canal-like system of membranes in the cell, and lysosomes, which is like the digestive system of the cell. So how energy is stored and used by the cell. So the mitochondria, which we just discussed, are present in a cell. They are the powerhouse of the cell, which synthesizes energy. Energy is stored in the form of ATP, adenosine triphosphate. Whenever cells need energy, the ATP is broken down to provide the cell with that required energy. Enzymes and inhibitors controlling metabolic reactions. So enzymes can be regulated by other molecules that either increase or reduce the activity. Molecules that increase the activity of an enzyme are called activators. Molecules that decrease the activity of an enzyme are called inhibitors. When a toxin molecule enters into the lipid bilayer and pores, the balance of the ion gradient's maintenance is disrupted. As a result, change in that intracellular water content can happen and cell death may occur. So in researching the water and why our fish have died off, I have looked into the Carinea brevis blooms of the dinoflagellate brev toxin. So large die-offs of fish can be caused by toxins called brev toxins from blooms of the dinoflagellate Carinea brevis. Brev toxins are a family of complex polycyclic polyethers that are produced by the red tide algae Carinea brevis. that grows in the Gulf of Mexico. So brevetoxins are often abbreviated PBTX, which derives from their former designation, Trichodiscus brevis, and their chemical structure is carbon 50, hydrogen 70, and oxygen 14. Brevetoxins are truly toxic. They have been responsible for many fish, mammal, and mollusk kills in the Gulf, and tainted seafood has caused widespread food poisoning in humans. This year saw an especially virulent K. brevis bloom, especially off the coast of Florida. So where does this chemical come from and how is it used? Red tides, also called harmful algae blooms or HABs, occur when microscopic algae multiply to higher than normal concentrations, often discoloring the water. So although more than 50 harmful algae bloom species occur in the Gulf of Mexico, one of the most well-known species is the Carinea brevis, which is the red tide organism. Specific impacts of this chemical on fish and humans. So blooms of the Carinea brevis are reported to be the cause of the brevetoxin in seafoods, and these are lipid-soluble and heat-stable cyclic polyether compounds. A number of BTXs have been identified. BTX2 type B is reported to be the most abundant in this group of toxin in K. brevis. BTX binds to voltage-gated sodium channels in nerve cells, so leading to disruption of normal neurological processes. Consumption of the toxic fish can cause tingling of the facial muscles, dilation of pupils, and a feeling of inebriation. What organs and tissues are affected by this chemical? Neurological signs and symptoms include paresthesia, reversal of hot and cold temperature sensation, vertigo, and ectasia. Inhalational exposure may cause respiratory symptoms such as cough, dyspnea, and bronchospasms. The brevetoxin can also cause illness in aquatic wildlife by affecting the central nervous system. Brevetoxins affect fish regardless of the species or developmental stage. Signs of intoxication in fish include violent twisting and corkscrew swimming, defecation and regurgitation, pectoral fin paralysis, caudal fin curvature, loss of equilibrium, and convulsions. Ultimately, fish die because of their gills. They just stop functioning. How does brevetoxin enter the cell? So they are a polycyclic ether, which we have discussed. They bind to and stimulate sodium flux through voltage-gated sodium channels in nerve and muscle. These toxins are depolarizing substances that open voltage gate sodium ion channels and cell walls, leading to uncontrolled sodium influx to the cell. What metabolic process is specifically impaired? So brevetoxins have been shown to undergo cellular metabolism through known xenobiotic biotransformation pathways for detoxification, primarily in the liver. When there are high concentrations of this algae, the water can become poisoned when the algae releases a toxin called the brevetoxin. The brevetoxin can be eaten or inhaled. This toxin affects the nervous system by opening the sodium channels on neurons. This can lead to paralysis and loss of coordination. So what caused this problem? So the K. brevis organism is typically found in the Gulf of Mexico and our coastal waters from a few hundred to a few thousand cells per liter of water. However, when it comes into contact with a major source of excess nutrients, it expands into a red tide bloom with at least 100,000 cells per liter to as many as 5 million cells per liter. Its growth from a major nutrient source is achieved by asexual reproduction, where one cell becomes 2 and 2 becomes 4, etc. There are a variety of nutrient sources that can sustain a red tide bloom, from natural sources like dead fish and other decaying sea life to man-made sources like air pollution and runoff. So air pollution, smoke from power plants, automotive exhaust, agriculture, mining dust are all main components of air pollution that fall into our coastal waters as excess nutrients. The runoff, effluent from sewage treatment plants and from waterside septic systems are a source of runoff that puts nutrients into our coastal waters, while red tide blooms typically form offshore. Monitoring by scientists has shown that the blooms intensify as they move closer to shore from the nutrients and runoff. Recent research has shown that leaf litter from trees and grass clippings are a major source of nutrients and runoff that contributes to the excess nutrients in our waterways. And then wind and ocean current flow. So periodic upwellings drive the nutrients up a water column in the ocean, where they are consumed by the K. brevis cells, causing them to multiply and initiate a red tide bloom. Ocean currents and wind drive the forming bloom towards our shore. As the bloom moves closer to shore, it is further enriched by the nutrients from our land and runoff. Here are my sources, and that concludes my presentation. Thank you, everybody.